Since the COVID-19 pandemic, probably a lot of us heard of the PCR test, but do we actually know what is the PCR test and what is it used for? In today's video, I'll be talking about the polymerase chain reaction, which can also be called molecular photocopying. What is PCR? Polymerase chain reaction is a fast and inexpensive technique used to amplify or copy small segments of DNA. Because significant amount of a sample of DNA are necessary for molecular and genetic analysis, studies of isolated pieces of DNA are nearly impossible without PCR amplification. What is PCR used for? Once amplified, the DNA produced by PCR can be used in many different laboratory procedures. For example, most mapping techniques in the Human Genome Project relies on PCR, which its main goal is to complete mapping and understanding of all the genes of human beings. In addition, PCR is also valuable in a number of laboratory and clinical techniques, including DNA fingerprinting, detection of bacteria or viruses, particularly AIDS, and another example would be coronavirus, since we do the test to see if we're positive or negative for the virus, and also diagnosis of genetic disorders. How does the PCR work? This is a simple summary of the procedure. Step 1, denaturation. Step 2, annealing. And step 3, extension. And throughout the video, I'll be mentioning them step by step. To start the process, we need buffer solution, PCR tube, nucleotides, which are the building blocks, adenine, thymine, cytosine, and guanine, Taq polymerase, primers, and the DNA template. If we're gonna think of it in a simple way, it's basically we're putting all the things the DNA needs to perform the replication and then letting the DNA replicate by itself. The first step is denaturation, in which double-stranded DNA templates are heated to separate the strands. Then, short DNA molecules called primers bind to flanking regions of the targeted DNA, and this step is called annealing. In the third step, which is extension, the DNA polymerase extends the 3' end of each primer along the template strands. This process results in the duplication of the original DNA, with each of the new molecules containing one old and one new strand of DNA. Then each of these strands can be used to create 2 new copies, and so on and so on. The cycle of denaturing and synthesizing new DNA is repeated as many as 30 to 40 times, leading to more than 1 billion exact copies of the original DNA segment. In addition, the entire cycling process of the PCR is automated and can be completed in just few hours. It is directed by a machine called the thermocycler, which is programmed to alter the temperature of the reaction every few minutes to allow DNA denaturing and synthesis. And thank you so much for listening. I wish this video was helpful and you learned something new.